Hello, thank you all for joining us today to hear about how you can use the CellRaft AIR system to enable your suspension cell line development. I appreciate you taking the time to tune in and hear about how we can help enhance your workflows and enable you to perform the best possible science. So if you're tuning in today, I'm sure you already know that cell line development is laborious and expensive. It all starts with a heterogeneous population of cells that you then have to take through several rounds of selection and screening, identify a single cell population, and then expand and evaluate your clones. There are a lot of pain points in this workflow in the traditional method. It requires dozens of consumables in order to capture those low frequency events, typically less than 10%. It's incredibly manual, and all of that screening and manipulation takes hours. And I've spent many of those hunched over a microscope, desperately hoping to find a cell line that I'm interested in, only to be disappointed at the end of weeks of work. Oftentimes, because of all of those selection steps and manipulations that you have to do for the cells, at the end, you're left with unhappy, perturbed cells that aren't really even phenotypically capable of giving you what you want. And ultimately, you have to use all of these multiple expensive pieces of equipment, such as flow sorters, high content imagers, next-gen sequencers, or real-time PCR machines. And after weeks and weeks of characterization, you might get to the end of all of this and only have one clone that's subpar or no clones at all. So I'm going to talk to you today about how you can use the CellRaft AIR system to save time, money, and improve the quality of the cells that you get at the end of your workflow. Our system offers numerous advantages that I'll go through in the talk today, including the ability to use a single consumable, the CytoSort array shown here on the left. Using that, you can screen thousands of cells at a time without using multiple consumables. The system itself is fully automated and hands-free, allowing you to perform imaging, isolation, and data acquisition in a completely automated fashion. Because our system is incredibly gentle and there's no manipulation of the cells required once they've attached to the array, we're left with hundreds of viable phenotypically stable cells that have not undergone successive rounds of selection. And you can perform the entire workflow from imaging to analysis to isolation using our single affordable instrument, the CellRaft AIR system shown here on the left. When you look into purchasing a piece of lab equipment to fulfill a need in your lab, there's a lot of different choices in this space to perform these single cell type workflows. At the high end is high content imaging, which like to do very beautiful bright field and fluorescent imaging. You can look at your cells in situ in the plates. You don't need a lot of cells to get started, but unfortunately you can't actually recover those cells at the end. So you're left with doing multiple rounds of cell selection or identification. We most often get compared to the flow cytometers and the droplet dispensers in this space because we allow you to image and isolate your cells. But unlike those instruments where you are needing large sample inputs, you can't actually phenotypically characterize your cells, and you need a lot of cells to get started, we think the AIR system really solves all of these issues that other technologies might have and allows you to combine the power of imaging, both bright field and fluorescence, with the ability to actually recover the cells that you have looked at in the dish. The workflow is incredibly user-friendly and very easy to get started with. There's an incredibly low barrier to entry, and most of our customers tell us that getting started with it takes a matter of days, not weeks or months. The whole process starts with the CytoSort array. The process begins by prepping the array to prepare it for cell seeding, and cells are simply seeded by droplet dispensing. Once the cells have attached, we perform that image and identification step using the CellRaft AIR system. And after your designated experiment is concluded and you've identified all of the cells that you're interested in, you can target them for isolation and retrieval. And then off the array and off instrument, the cells can either continue to grow off of the raft as shown here in 96 well plate for clonal expansion, or you can assay them directly for lytic endpoints like sequencing or viability assays, if you will. So to go into a little bit more of the technology, I like to describe the CellRaft and the CytoSort arrays as sort of a new way to do cell culture. It has a very familiar form factor and it does look and feel like a cell culture dish, but it's a very unique way to do a lot of single cell culture using just a single footprint. The CytoSort array is comprised of an elastomeric microwell array shown here. Within each small microwell, we fabricate a releasable magnetic polystyrene cell raft, which is the surface that the cells can attach and grow on. Unlike a lot of other plating methodologies where you have a single lonely cell sitting in a large volume of media as it tries to grow, one of the benefits to the cytosort array is that there is a contiguous volume of media that all of the cells share. So even though you have single cells in isolation in the microwells, that contiguous volume of media enhances cell viability, allowing the cells to share paracrine factors and ultimately be happier in culture. Despite the sort of gold color that you see in the bottom of the array on the far left, the rafts themselves are optically transparent, which means we can use both fluorescent and bright field microscopy to perform very high resolution imaging of every single raft in the cytosort array. What this allows us to do is perform phenotypic-based imaging, analysis, and isolation of single cells or colonies in your experiment. We have the consumable available in a variety of different flavors. I always call them flavors now because one of our associates says they look like waffles, and that's something you just can't unsee once you've heard it. So you can see here what the arrays look like. They come in several different types. We have them available in the raft size of 100 by 100 micron square, 200 by 200 micron square, and then 500 microns by 500 micron square, which will be releasing next month. The pros for the 100 by 100 is this is sort of the most bang for your buck in terms of single cell screening. This has the most positions available to it and allows you to look at the most number of cells. The pro for the 200 by 200 is that you can grow larger colonies. So if your cells are slower growing or more spread out, or you have fewer of them, you can grow them to slightly larger sizes prior to isolation. And the 500 by 500 rafts are meant for three-dimensional growth. In terms of performance, the 100 and 200 arrays perform pretty much the same. We typically expect around 20 to 30% of the cells that we see to attach as single cells. But fortunately, because of that improved viability, almost all of the cells that we see attaching as singles will ultimately form a colony on the array. Once we isolate those formed colonies that have grown on the array, we expect outgrowth of your typical cell line to be greater than 85%. So the majority of what you isolate will grow into a viable colony at the end of your experiment. In addition to choosing the size of the raft that's appropriate for you, you can also choose the format of the array itself. They currently come in three different formats: the single, which has one central reservoir, the quad, which has 4 individually spaced out reservoirs, and the hexaquad, which has 24 reservoirs. And depending on your application, you can choose the right consumable for your experiments. For example, the quad is particularly well suited to performing assay validation or cell line validation, and you'll see that a lot in today's talk. The trade-off here is more variables, but you have fewer rafts for interrogation. So if you're really looking to screen thousands and thousands of cells, the single reservoir is a better bet. Ultimately, we recommend that you seed your arrays with around 30 to 70% of the cells to available positions. It's very cell line dependent. We recommend that you optimize it, but typically you can screen on the order of thousands to tens of thousands of cells using just one of our consumables. The ease of use really can't be understated for the cytosort array. If you've ever seeded cells for a typical cell culture experiment, you'll have no trouble getting familiar with our technology very quickly. The workflow begins by preparing a low concentration cell suspension at your desired concentration. The cells are manually pipetted into the array and are allowed to settle by gravity into the microwells. We don't directionally put the cells into the microwells themselves. They'll just settle by a statistical like distribution. And once they find a raft, they will attach to it and grow on the polystyrene cell surface. Once you've seeded your cells, the nice thing about the consumable is that they just go into any standard tissue culture incubator. You don't need to keep them on the instrument itself, and they can just grow as many arrays as you would like at 37 degrees at a single time. When you're ready to scan your arrays to look for single cells or clones, the cytosort arrays go on our CellRaft AIR system shown here on the right. The AIR system has a bright field and 3-color fluorescent inverted microscope with a 10x magnification on board. All of the image acquisition and data storage is fully automated and occurs completely hands-free, which is really quite nice. So there's no risk of losing your data or a file not saving or not capturing your data if you're not standing there at the instrument. Once you've identified your cells of interest for retrieval, you can map your cells for isolation and perform automated release and transfer using the AIR system. You can see here in the blowout of the AIR on the stage deck, the array sits on the left side and the collection plate sits on the right. We can perform that entire workflow, image, analysis, and isolation in as little as 30 to 45 minutes. That 30 minutes is our newest iteration of the AIR system with an upgraded isolation and retrieval. The AIR system does not currently come with environmental control, but it can be upfit with an optional stage top incubator that provides environmental control, including temperature, humidity, and CO2. If the AIR system is the brawn, the proprietary software is the brains of the whole operation. So we have developed our own software internally that drives the automated portion of the AIR system. You can use this software to perform all of your imaging and data acquisition. It's an incredibly user-friendly interface. It's very easy to get started with, and there's lots of different options that you have to interact with the software and the AIR system. You can use the software to perform real-time imaging. So you can basically look at your arrays in live scans without any image capture. You can toggle colors or imaging settings such as binning, gain, and exposure. However, the real power of the system comes not in that real-time imaging, it comes in the automated full array scan. Just by clicking one button, you can set up the array to have a full scan from top to bottom. It will scan every single field of view and every raft in that field of view, which means that we can capture really impeccable time course images of your plates as they grow over time. And this is one of my favorite aspects of the AIR system. If you've ever tried to use software to look at cells in a 96-well plate or even on a microscope in a high content imager, the cells move all over the place and you can't look at the same cell twice. That's not the problem that the AIR system has. You can follow the exact same cell over time every single time you scan the array, and you'll see some examples of that later in the talk. You can also zoom in and look at cells of interest in greater detail. If you have a fluorescent marker or you're looking for a phenotypic characteristic of choice like morphology or growth rate, you can do that type of imaging. And we can also zoom in and look at these different fluorescent colors and determine whether or not the cells have the phenotype of interest prior to isolation and selection. That ability to serially image the array means that we can actually look at every single cell over time, and we can play these little movies right there in the software in order to determine whether or not the cells are growing appropriately and forming a clone that you're actually interested in. Once you have found clones that you like and you would like to isolate them for downstream analysis, it's very easy to select them for further isolation. You can either directly map them right from the navigation software as you find cells that you like, or you can analyze your data using our next generation Cytometric software, which I'll show you later in the talk. So we've talked a little bit about how the whole process works together. So how do we actually get the rafts out of the array? You can see here the principle of this illustrated in cartoon. Because the rafts within the array are releasable and have been doped with magnetic nanoparticles, they can be retrieved simply by using a magnetic wand. You can see that illustrated here. So on the deck of the instrument, the raft that you have identified for retrieval is dislodged from the footprint of the array using a release needle that sits underneath the array. The magnetic wand dips into the fluid, picks up the raft for retrieval, and then takes it over to a 96-well plate for deposit. You can see here that the cells are now intact in the 96-well plate and then can grow off of the raft for further propagation. You're not limited to picking just into 96-well culture plates. You can also pick into PCR strip tubes or PCR plates if you're going directly to downstream lytic assays. To show you what this looks like in actual real time, you can see here on the left side what the CellRaft release looks like if you keep your eye on the raft in the blue box. What happens is that the raft is poked free of the array. The wand comes down and picks it up, and then the wand will pick it up, move it over to the collection plate for deposit, and then your cells are safely moved over to the collection plate of interest. So I like to compare this to popping an ice cube out of an ice cube tray rather than melting the ice cube, for example, like you would with the flow sorter. It's incredibly gentle, very efficient, and your cells never have to see trypsin again once they have been plated on the array. So I've talked to you a little bit about the principles of the system and the technology, and this slide is just to highlight really that the AIR system allows you to perform unlimited applications. As a tool company, it's important to us to really use the AIR system to enable lots of lines of study as opposed to just being a unitasker. And the AIR system really is the ultimate multitasker. Our customers have leveraged the technology for single cell omics, gene editing in primary and cell lines, three-dimensional culture, and cell-based assays. For the purpose of today's talk, we're going to focus on cell line development, and particularly the suspension cells. So we've spent a long time showing the value of the AIR system for adherent clonal cell line development. And there are a lot of advantages for using the AIR system for this purpose. We can have a really beautiful track and traceability from your single adherent cells to clones because they attach to the surface of the array very nicely. You can use the consumable to screen thousands of cells and the cells don't move around. We can use less media and plasticware because we're only picking clones that we have already shown to be viable and have grown from the single cell state. And over 70% of single cells ultimately form colonies for the most part. So what I'm showing here in these graphs is an experiment using adherent CHO cells in comparison to limiting dilution. So you can see with limiting dilution, you don't really even know how many wells you're starting with have a single cell. We plated 10 96-well plates, and we were able to retrieve 27 clones out of 10 96-well plates. In comparison, using either of our formats, the CytoSort Array 100 or the CytoSort Array 200, we're able to screen thousands of clones. So rafts that had a single cell from the CytoSort Array 100, we had over 7,000 single cells, and over 5,400 of them turned into single cell derived clones on the array itself. As I mentioned before, the trade-off with the CytoSort Array 200 is the number of rafts that you have available to you because they're bigger. So there are fewer clones here, but we had over 1,400 single cells and over 1,100 of those turned into single cell clones. After isolation, we isolated 3 96-well plates per condition. And again, you can see there's those 27 limiting dilutions clones. And in comparison, there are well over 200 clones for both of our array formats. So there's that performance that we see, which really, it doesn't really matter for some cell types, which size you pick. But the great thing here is that using this workflow, you have hundreds more clones to screen, which means you're going to succeed or fail much faster than you would in a typical limiting dilution experiment. And we're able to confirm that these were indeed clonal cell lines that met all of the criteria that we needed them to meet prior to isolation. So the question that we get asked all the time is, how do I leverage the CytoSort arrays and the CellRaft AIR system for suspension cells? And it's not a trivial question, actually. While adherent cells have been our bread and butter, and we're really good at doing that, we know there's a lot of interest in using suspension cells, particularly in the production space for antibodies or cell lines for therapeutic purposes. And it really sort of got us thinking how we could adapt the process in order to enable suspension cells on the array. Because they do grow in suspension, and one of the principles of the CellRaft system is that the cells have to attach, we needed to find a way to adhere the cells while still allowing them to grow in suspension after they had returned to bulk culture. To begin to determine whether or not this was possible, the first thing we wanted to do was to optimize the ability to coat the array. We have shown in the past through some of our other workflows that while these may look and feel similar to traditional cell culture consumables, they do require a little bit more finesse, and they do require a bit more extended coating than your typical T25 or a 96-well plate. So what we did was optimize the coatings using our CytoSort arrays by coating with substrates at 37 degrees for either 4 hours or 16 hours to determine the effective time. And we used 3 different commonly used cell culture substrates: poly-D-lysine, poly-L-ornithine, and poly-L-lysine, PDL, PLO, and PLL. This is one of the beauties of using the quad array. We can optimize these coatings or our cell types very easily just by putting something different in each of the reservoirs. So I really like to use the quads for this purpose because you don't have to use that much reagent and you can get a lot of answers with just that one format. We seeded 5,000 Jurkat cells per reservoir. These grow purely in suspension and they're really hard to attach. And then we scanned the arrays 4 hours post-cell seeding and 24 hours post-cell seeding to determine the number of single cells that were attached at all the time points and whether or not those cells would remain attached over the first 24-hour period. What you can see here on the left is that after 4 hours of coding in the blue bars, even in the uncoated wells, there in uncoated reservoirs, some of the cells do actually attach at 4 hours, but we saw an improved attachment with the cell coatings PDL, PLO, and PLL. But we were a little bit disappointed that the cell seeding percentage was quite so low. But when you look at the 16-hour time point in the orange bar, we get much closer to that target that we expect with our adherent cells, which is around 15 to 20% of our single cells going down to singles at that 4-hour post-scan time point. So what this tells us is that you need to coat the arrays longer in order to ensure maximal cell adherence in that first 4 hours after cell seeding. If you're going directly to single cell isolation, 4 hours might be sufficient for you because you're not letting the cells sit there very long, and you could certainly do that. But this sort of gives you a range of where that optimal coating time might be. Importantly, what we also found was that the additional coating of the arrays, both at 4 hours and 16 hours, really did improve cell retention. So what we're looking at here is the number of those single cells that we identified at 4 hours that were still there at 24 hours at that 24-hour scan. And what you can see on PDL is that in both the coating situations for both time points, greater than 90% of those single cells were still attached 24 hours post-cell seeding. The uncoated, obviously only 30% of the cells that we attached were still attached at 24 hours. So this gives you a really good idea that the coatings are completely necessary to adhere those single cells to the array itself. So in order to extend this, we wanted to test a range of suspension cell lines in order to determine whether or not this was widely applicable to a variety of different cell lines. And you can see here that list here. We tested the 293 freestyle cells, which are a suspension adaptation of adherent 293s, MAB 108s, which are a mouse hybridoma B lymphocyte line that's semi-adherent, and then the Jurkat, Raji, and THP1 cells, which are all completely suspension and do not attach to normal cell culture substrates. In this experimental design, we coded cytosort arrays, 100 quad cytosort arrays for each of the cell lines with either uncoated PDL, PLO, or PLL overnight at 37 degrees. We seeded 5,000 cells for each of the reservoirs, and then we scanned the arrays 4 hours post-cell seeding to get our initial time 0 to determine single cell attachment, and then every 24 hours until isolation and retrieval. We analyzed the single cell seeding and clonality using the next generation off-the-air software, and then isolated those clones of interest for downstream expansion to determine whether or not they could go back into suspension culture. So we were quite excited to find that the suspension cells actually do adhere and divide on the coated arrays. And what I'm showing you here is 3 different cell types, the Jurkats, the MAB 108s, and the THP1s. And there are a few things to highlight on this slide. The first of which is just really how beautiful the imaging is. Even though it's only 10x, we can expect around a 20x magnification because of the quality of our camera. And unlike any other consumable I've ever used, there's just a really amazing ability to verify that you have a single cell in each raft. This is particularly easy with these coatings because the cells stick down and they attach and then they never really leave again, which is really quite delightful. It is also very exciting that the cells not only remain attached, but the daughter cells remain attached as well. So if you look at the Jurkats and the coatings, you can see that 1 cell turns into 2 cells turn into 4 cells, and 4 cells turn into 8 cells. So this time course imaging allows you to very nicely monitor growth rate to ensure that you haven't selected a clone that is phenotypically different than your population, or that you're actually getting cells moving into your rafts, because if you start to see time points that have 30 cells or 40 cells or no cells, you'll know that you've lost your clonality. The other nice thing here is that you can also monitor what your clone looks like on the day of isolation. So unlike flow sorting or droplet dispensing, where you have really no idea what goes into your well, we know exactly what these clones look like on the day that we isolate. And for example, in the jerkouts as well, at day 4, we can see that in the PDL, PLO, and PLL coatings, you have a nice 8-cell colony when you isolate. In comparison, by day 4 on the uncoated, even though you saw some cell growth and attachment, by day 4, that clone has detached and is no longer present. So it lets you know that your cells are still there, that they're still healthy, and that you haven't lost the clone because they have detached from the surface. I also want to highlight the THP1 cells. You can again see that really nice attached morphology of the cells growing in the plane and all of the daughter cells still attaching. In comparison, when you look at the uncoated well at day 4, you can see that the cells are starting to ball up, come off the cell surface, and my guess is that clone would not have survived the isolation process because it would have detached during isolation and retrieval. So using this time course imaging and taking pictures every single day, you can single cell verify, you can get a data record of your clone from start to finish, which is really important for all of your pharmaceutical applications, particularly in the gene therapy space. And of course, you can monitor phenotype, doubling time, colony growth, or really anything that you can do with an image you can do using the AIR system. This experiment also really highlighted to us how critical it is to optimize your cell lines prior to embarking on any experiment using our system. This is something we always tell our customers or potential customers that it really is important to understand what your cell type does in our system. The 293 freestyles shown here, they actually attach really, really well, even in the uncoated scenario. So you can see up here at day 1, there's a single cell, and by day 4, you have a nice adherent clone. In contrast, PDL, which has been a workhorse for us for all of the other suspension cell lines, was completely toxic to the 293s. If we hadn't done this optimization, I would never have known this and could potentially have just killed all my cells for no reason. PLO and PLL had less of an impact, but you can still see in both of those coating situations, the morphology does not look right for these cells, and they're growing much slower and starting to detach. So this really highlights to us the beauty of the quad, because you can optimize these conditions as well as understanding your cell line prior to embarking on any critical experiments. So this was a lot of data, and it was important to us to use all of this data in a thoughtful way in order to really show that we can attach these single cells and recover them. And fortunately for us, we've spent a significant amount of time really revving our Cytometric software that allows us to analyze the cells and clones that grow on the arrays. We have implemented some really advanced bright field analyses that allow us to look at a lot of different metrics. What you can see here is a screenshot of the new user interface. It allows you to define unique subpopulations. What I'm showing you here is the criteria for looking at single cells for the jerkouts in this experiment. You can select the scan to interrogate, which reservoirs, the segmentation method that you want to use, as well as toggle a variety of criteria in order to optimize the analysis parameters for your specific cell type, which is really quite amazing. So using this setting, we can define what a single cell means to us for the jerkout cells and the THP1 cells, or really any other cell that we are interested in. If we are looking for clones, you can use a different metric that we call coverage. This allows you to really get a very nice perspective on how many cells are in each of your rafts. We call it coverage rather than confluence because we're really looking for the amount of the raft surface that is covered by your cell population. This is a super easy and useful way of determining whether or not you still have a clone. If you can imagine starting from a single cell and in only 3 or 4 days, there's no way that we would have gotten to this many cells. It's just way too many. So you can use the coverage metric to determine optimal raft coverage for clonal development, as well as for ease of isolation and transfer. But the best part of the new software is the ability to combine all of these metrics into a really powerful subset. So what you can see here is a subset on the left where I've set 3 different criteria, and I've been able to find the intersect of those criteria to very intricately define exactly the cells that I want at that specific time point for isolation. In this particular example, I have subset to look for single cells at time point 0, cells that doubled at time point 2, and cells that were confluent at time point 4. And you can see that list here in all of the cellular features, as well as get a whole raft view of the cells in each of the clones that are within that population. When you've identified those subpopulations for mapping, it's very easy to map them for isolation directly in the software. What you can see here is a heat map of all of the rafts that met the criteria that I selected in the previous slide. And you can see that that top left quadrant, that top left reservoir is the uncoated, and the bottom three are the coated reservoirs. And I think this is a really nice way to highlight your phenotype. And for example, in this particular instance, that the uncoated reservoir does not retain single cell derived clones, whereas the coated reservoirs do. Once you've found those rafts, you can simply click into the plate and map them for isolation. So using the software, we were able to identify which of the rafts actually had a single cell 4 hours post-cell seeding. And again, this is very cell independent, but the general trend is that coating the rafts does give you more single cells and a higher percentage using the coatings for every single cell line, with the notable exception of the 293 freestyles. Importantly, even though there were a similar amount of single cells in the uncoated arrays at 4 hours, simply due to gravity settling and transient attachment, if we looked 24 hours post-seeding, you can see, particularly for the Jurkats, the Raji, and the THP1 cells, there's a dramatic retention of the cells in the coated as compared to the uncoated. So with the uncoated reservoir, only about 20 to 30% of the cells were still there after 24 hours, whereas over 90% were there for the coated reservoirs. And again, the 293s are the outlier here. Uncoating was actually just fine for them, and the coatings were actually more detrimental than helpful. So after 4 days in culture, we were able to isolate cells for retrieval and downstream analysis. We looked 4 days using the same software. We wanted to see exactly how many of those single cells had turned into clones. And what you can see is that for the most part, we get about 50% clonal formation from those cells that were singles, depending on the cell line. Some are as high as 90% in the Raji cells. And again, it is fairly coating dependent. But the key takeaway here is that by simply coating the arrays with standardly available cell coating substrates like PDL, PLO, and PLL, we are able to generate hundreds of single cell derived clones using just one reservoir of one quad array. So for example, if you look at the Jurkats here, we had about 82 in the uncoated, but there are well over 400 in the PDL and 300 in the PLO and PLL. So this really does give you just an abundance of wealth. It's more clones that you could ever hope to characterize in a single experiment. But the best part about this is that you can then pick the clones that really are the best of the best. You don't have to settle for whatever sad clones grow out of your poor limiting dilution experiment. You're actually able to ensure that the cells meet all of the criteria for success for you, whether it's gene edited or fluorescent or non-fluorescent or a particular growth rate or morphology. The coating preference is fairly cell line dependent, so I don't want to generalize as to which coating goes to which cell line, but you can see here that some of the cells really don't seem to care, and some like one better than the other. What I'm showing you in this graph is the ability of the clones to grow off raft once they've been isolated. So this is a key metric for us. Obviously, it's no good if the cells remain attached to the raft and can never go back into suspension. So this graph quantitated the percentage of rafts that we isolated that actually allowed the cells to go back into suspension and grow off raft and form a suspension cell clone. And we were really quite gratified to see that over 75% of the rafts that we isolated did in fact form a viable clone for downstream propagation or analysis. The coating, fortunately, does not inhibit colony growth either on or off array. And we do see a very high percentage of isolated clones being propagated off raft. To show you what this looks like in bright field imaging, these are all clones that were isolated off of the coated rafts. You can see that the cells themselves are detached from the rafts or growing just off the rafts and have gone back into form nice suspension clones. Again, with the notable exception of the 293 freestyles, which are happy to grow adhered to the CellRaft surface. This is fixable, however, they can be easily dislodged from the surface just by gently pipetting the culture media in the 96 well plate. So we've been able to show that we can grow suspension cells off raft with high viability. They can return to suspension cell culture very easily with zero manipulation, and then they can be passaged normally for subsequent clonal expansion or analysis. So through this process, we have developed some recommendations for success if you're thinking about using our technology to enable your suspension cell workflows. First and foremost, I can't emphasize enough how great it is to use the quad arrays in order to do efficient cell culture condition optimization for new lines. Whether it's cell number or coding substrate or cell type, it really does give you a lot of flexibility and power to optimize your experiments very quickly. We also recommend that you serially image the cytosort arrays every day, or at least once a doubling time, in order to monitor that single cell starting point, growth rates, morphology, and clonality. Particularly with the suspension cell workflows, you're obviously at a risk of losing cells or cells moving, and having that ability to monitor cells at every single time point does give you confidence that you have a monoclonal cell line at the end of your experiment. The next generation off-the-air software is incredibly powerful. It gives us the really unique ability to be able to identify single cells very easily, determine colony growth at multiple time points, and then map those plates for downstream isolation. And really, we recommend that customers isolate just as few as 196 well plate of CellRafts, because you can really expect up to 90% outgrowth efficiency. It's a really common question that I get, how many plates can I isolate in a day? How many clones can I isolate? And everybody always says, I want to isolate 300 or 400, because that's what they're used to doing in their flow-based or limiting dilution-based workflows. I guarantee you, if you isolated 400 clones, you would get 350 of them to grow off raft, and then you'd be left trying to decide which of them are valuable to you. So you can really isolate as few as one plate and expect hundreds of clones that most likely will meet your criteria and will continue to grow and propagate in culture. So to summarize what I've shown you today, it is totally feasible to coat the cytosort arrays in order to enable suspension cell line development. And we think this will be really powerful for individual groups that are looking to use it for antibody production or protein production or patient cells. You can generate hundreds of characterized clones in as little as 1 week. This entire workflow from seeding cells to growing clones took about 10 days. And really the possibilities for what you can do with those cells are limitless. You could CRISPR edit your cells prior to seeding, you could perform omics, you could do on-array or off-array drug screening of your clones, and really have an abundance of material in order to get started with your downstream analyses even faster. So suspension cells are really just the tip of the iceberg for us. Cell line development is really important to us, and it's something we're really good at, including CRISPR editing. But the great thing about the AIR system, as I mentioned, is that we've been able to show a variety of different applications can be effectively leveraged on our system, ranging from induced pluripotent stem cell differentiation to genomics to cell-based assays to antibody staining on array. And if you're interested in any of those other topics, we have a lot of materials on our website, or you can contact us for more information about your specific application and how the AIR system might be able to help you. And we're really excited about our next product, which is launching our organoid bundle on February 7th at the SLAS meeting. We're booth 432, so if you'll be attending that meeting, please feel free to come by and see us. But that bundle is including the new consumable I briefly mentioned, as well as some analysis software to enable 3D workflows, and as well as hardware and software upgrades to the system to enable 3D culture on the array. So with that, I will conclude, and I'm happy to take any questions from the audience that you might have. Okay. Well, if there's no questions, then feel free to message us or email me or contact me offline, and I'm happy to talk to you about your specific workflows in great detail.